everybody, and welcome to this week's Lunch with the Scientist. Today, we are joined by Dr. Cynthia Corbitt. She has a broad range of training in neuroendocrinology and physiology, including processes and disruptions of sexual differentiation, and specifically in anatomical, molecular, physiological, and behavioral techniques. Her research focus has been on environmental signaling. So I'm going to go ahead and welcome Dr. Corbitt to the stream. So welcome.
Thank you.
So we'd like to start out by hearing a little bit about your path and your story of how you became a scientist. I might ask you a couple of questions here and there. So sorry if I interrupt, but I'll try my best to have a natural little entryway.
No problem. Well, I have some slides for that.
Perfect. So we'll go ahead and get started.
Okay, well, I'm currently a professor in the Department of Biology at the University of Louisville in Louisville, Kentucky. Hopefully my Kentucky background is showing up. So I have been here since 2002, which just hit me not too long ago that's 20 years, which is a long time. But a little bit about how I got here. I started deep in the heart of Texas. If you're in Texas right now, you should be clapping your hands 4 times. So I am from Houston, which is where I was born, grew up, went to high school. I did my undergraduate degree, a Bachelor of Science in Biology at the University of Texas at Arlington, which is up here between Fort Worth and Dallas. So once I had finished my bachelor's degree, I moved all the way up to the far north, Fairbanks, Alaska, where I did my PhD in biology. From campus, we could see Denali, which is the highest point in North America. During the winter, we could see the Northern Lights, which is spectacular. And I did most of my work in this boreal forest that's shown here, where I studied little songbirds called dark-eyed juncos. I see them around my area in Kentucky in the winter, but they spend their summer in the far north. And when I would go look for them, I would actually listen for them, because when they sing, they have a song that sounds kind of like a cell phone. It's like, and I would just listen for cell phones in the middle of the forest. And that's where I would typically find them, up at the top of one of these spruce trees. So that's what a dark-eyed junco looks like. And what I did for my doctoral dissertation, getting my PhD, was look at the effects of photoperiod or day length and a neurochemical called opioids that the birds make in their own brains, and look at the combination of those effects on singing in the dark-eyed junco, which the scientific name is Junco hyemalis. So that's me around 1994. Catching a bird. I don't know if you can see that in front of me, I have a mist net. It's very thin. The birds can't see it, but I put a audio recorder right next to the net that has a cassette tape, we used those back then, of a dark-eyed junco singing, and the resident juncos would get super mad, and they would come zooming down out of the trees, and they would get caught in the net, and then I could catch them and take a blood sample and measure things like their testosterone, their sex hormones. So I have also brought these birds into captivity and studied things such as mate choice and what factors make birds choose one mate over another. So this is a female dark-eyed junco checking out 2 different males and trying to decide which one she wants to hang out with. And part of that decision is based on how they sing. So these are little snippets of different songs that different male dark-eyed juncos sing, and what it actually looks like on a sonogram when you put the frequency of the song versus the time. And even though to my ear, and yours probably too, these sound very similar, just repeated notes. It sounds like, to the female, they can tell these different note types apart. And I also studied the brain region. So this is a brain of a junco that has been sectioned from front to back, put on slides, and stained with a stain so that I can see this region right here called the HVC that controls singing in these birds. So that's the work I did as a young student, getting my PhD, and then moved on to do postdoc. So I don't know if you know what a postdoc is, but a postdoctoral position is what you do after your PhD. You're still considered to be in training, but you have a little more independence and you're supposed to be running your own research program. So remember I said I looked at opioids in birds. Those are just the neurochemicals that have something to do with motivation. Well, I was looking at those same chemicals, but this time looking at maternal behavior in rats. So I changed models and I really changed location because I did this at Tufts University Medical School, which is in downtown Boston. And was there for 2 years, ended up moving down south to Tulane University Medical School, where I did a second postdoctoral position funded by the National Institutes of Health. And this time I learned about endocrine disrupting chemicals, which became a theme for most of the rest of my career, looking at compounds that mimic hormones or disrupt hormonal signaling. And that's that environmental signaling component that Jen mentioned. And this was at Tulane University Medical School at the Center for Bioenvironmental Research. So I started deep in the heart of Texas. That's where you clap 4 times if you're from there. Went to the land of the midnight sun up in Alaska, worked my way over to Boston, which I love Boston now to visit, but I didn't really care to live there. Moved to New Orleans, where I really miss the music now that I am here in the middle of the country in Kentucky and Louisville, which has worked out really well. So that's my path. I can talk a little bit more if you have any other questions, Jen, about why I chose those things, but that's what I did.
Yeah, I did have a quick question because moving from birds to a mammal like a rat is a big change. Why did you decide to make that progression?
Part of it was that I was a little bit constrained by I needed to be in Boston for family reasons. And so I had to find a location, I had to find a lab that would accept me, where I could write a grant to fund my postdoctoral fellowship years. And the person that was the best fit for me, because of the opioid connection, I didn't talk about it too much with the birds, but half of my dissertation was looking at these chemicals that act like morphine, that affect motivation, that affect the singing in the birds. Well, Dr. Ron Hammer at Tufts was looking at that same neurochemical, but how it affects the motivation to care for offspring in rats. So it was really the neurochemical that brought me to rats and not the fact that I wanted to look at rats.
So do you think that it made a difference in the wild birds that you were looking at versus a rat, which is a little more controlled in a lab in your results?
Well, I think some of the basic neurochemistry is really similar, and the receptors that they work on in the brain work a lot the same way. Of course, the brains are very different. But in both cases, we're somewhat looking at motivation.
Great. Awesome. All right. Well, I'm going to go ahead and let you continue. Those are just 2 questions I had.
Okay, sure. That was a natural place to stop me. Okay, so now I'm moving on to what I've been doing over the past few years in what I call the CAN lab, as in we can do it, but it's based on last names of the main people working there. So me, Cynthia Corbitt, Mikas Abelins-Abels, and Rachel Neal. So some things that we've been doing over the past several years, I'm only going to touch on details of a couple of them, but been looking at social dance and how it might help wellness in people with post-traumatic stress disorder. Been doing that with an organization called Dancing Well, the Soldier Project. I'll talk a little bit more about that in a minute. A project that's been a few years now looking at prenatal exposure to valproic acid, which the brand name is Depakote. It's an anticonvulsant, and looking at how basically what are the factors that lead to increased incidence of autism in males. And so some hypotheses are that, the testosterone difference, having more testosterone during development might increase vulnerability. Well, valproic acid is a model that is used in mice because it increases vulnerability in women who take it for seizures when they're pregnant, especially in their male offspring. So we were looking at valproic acid, testosterone, and maleness, and a mouse model of autism. One thing I'll point out that is a similarity to my current work is it's a prenatal chemical with a postnatal change, in this case testosterone, and then also looking at sex differences. Have a side project that really is headed by Kira Taylor and a former student of mine, Monica Unseld, looking at tear gas and menstrual cyclicity. Here in Louisville, we did have a lot of protests and disruptions back in 2020, and tear gas was used on the protesters. So we're looking at how that might have affected reproductive cycling. Dr. Abelins Abels, who's the of the Khan Lab, he's looking primarily at songbirds, stress. So I was so excited to have him join the lab because of my former work. I hadn't worked in birds in a long time, since, Alaska. And so he is bringing songbird physiology back into the lab. And so it's fun to play with those projects. Very recent project with Rachel Neal is looking at, again, prenatal exposure to vaping. So when pregnant people smoke, how does that affect their offspring? It's touted as being safer than cigarettes, but some of our studies using mice are finding that, even if it might be safer in some respects, it's still not safe. We are finding some effects on the offspring. And the main project that I'm working on now, also with Dr. Neal, is looking at prenatal cigarette smoke exposure. So when pregnant people smoke cigarettes, how does that affect the gut microbiome, the combination of species of microbes in their guts, their metabolism, and their behavior? So I'm going to talk a little bit more about three of these. So the first one is in collaboration with Deborah Denenfeld, who is the founder and executive director of Dancing Well, the Soldier Project. She's looking at post-traumatic stress disorder and traumatic brain injury in veterans. It's reported that 10 to 13% of U.S. veterans experience PTSD, often combined with traumatic brain injury from their time at war. And one problem is that they often isolate themselves at home and avoid going out and being in the community because there are so many things out there in the world that can trigger some of their symptoms. So what Deborah has founded is a weekly social dance in a safe place. We turn off the overhead lighting because some of the flickering can be triggering. We use lamps everywhere. We basically just form a community where people feel safe coming, and they learn how to do community dance. So if you've ever done square dancing, as part of your school elementary days, it's a lot like that. And we have a live band with a piano player and a violin player. And we wanted to see if participating in this group could be shown to increase wellness, as measured by several different psychological measures. And the take home there is we found that it did. And so what I'm showing you here is of the people in the study with PTSD who took 3 different psychological surveys before starting the program, and then took the same surveys 10 weeks later after being in the dances once a week. What we found is this is individual data points And then the average goes from about 100 probably to about 125, but it's statistically significantly increased overall, that people who participated felt better. And, how do you define wellness? Well, feeling of connectedness, decreased feelings of suicidality, and so forth. And, part of the program, it could be, part of it is the dancing itself, the physical activity, but there's also touch in this kind of dancing. As you can see, we're doing a promenade around the circle, and people touch hands. We have snack time in the middle of the dance session where you talk and chat about your life. And if someone doesn't come because they're isolating, well, Deborah calls them and says, we missed you. We miss you when you're not here. So all of these things work together to increase this feeling of wellness in the people who participate. And this kind of outcome is helpful. And this was totally blinded. We did not know who was answering the questions when the studies were scored. It helps Deborah to get more donations and more funding so that she can keep putting these types of programs on. Unfortunately, they did have to stop during the pandemic, but they are starting up again, so that is good news. Next, a little brief foray into hormones and behavior in songbirds, which is led by my colleague, Dr. Mikas Abelins-Abels, who's new faculty here in the department. And so really what he's looking at is a model of nest parasitism, where there are these nest parasites that will lay their egg in the nest of another species. And that other species either rejects the unfamiliar egg or takes care of it, but then they have a cost in terms of their energy raising another species young. So he's interested in interaction of stress, physiology, environment, hormones, in looking at all of these behaviors. Now he has moved his work into looking at local environment, and he just got funded. Actually, I couldn't have announced it if I had recorded this a week ago, because he just got funded by the Center for Integrative Environmental Health Sciences to look at these birds as a model for pollution in the urban environment. And what he can do is put heart rate monitors in these eggs and actually monitor, well, he puts a fake egg with a heart rate monitor, but the birds will then give a signal of how their heart rates differ in polluted versus non-polluted environments. He has a TEDx talk, but I mention this here again because I'm so excited to have someone working on songbird physiology in the lab again after I hadn't done that in so long. So I'm really looking forward to taking part in that. And now to the big study that we've been doing and that we're about to jump into again is looking at effects of prenatal cigarette smoke exposure. So we know that prenatal exposure to cigarette smoke is not great for developing fetuses. Some of the known effects are preterm birth, premature, having a low birth weight, increased risk of sudden infant death syndrome. There are some neurobehavioral effects, which is why I first got interested in this. Some differences in things like anxiety-like behavior. And so what we have just been finishing is looking at pregnant mice who get cigarette smoke exposure throughout gestation. Now, you might imagine we're handing them a cigarette and they're drawing on it. That's not how it works. They're in a little cage. We have a machine that puffs on the cigarette and then blows the smoke into the chamber where they are, and they inhale that. And so once the offspring that are exposed to that only in utero during gestation, once they are born and start growing up, we can measure things like their gut microbiome, which is the combination of species that are in their guts, short chain fatty acids, which are metabolites of those gut microbes, and different aspects of their metabolism and their behavior. And then we've added an extra factor in this one study that we put some animals on a high-fat diet in adulthood versus a low-fat diet, because it's common that people who were exposed to cigarettes during gestation, especially in the US, would also adopt a high-fat diet later on. So we're wanting to look for sex-specific effects of this prenatal CSE, cigarette smoke exposure, and postnatal diet. So here is a brief flow chart of what we're doing. So we take females that we've mated, we expose them to cigarette smoke or room air for 6 hours a day throughout gestation, feeding them a regular fat diet. We then let them have their litters, and we're collecting tissues at weaning, which is postnatal day 20, 21. Again, at postnatal day 50 when they're becoming adults. And then at postnatal day 50, we split them into a low-fat diet, which is very low fat, or a high-fat diet, which is not super high fat, but it's, getting there. And then we collect tissues at 3 months old, study their behavior, and collect tissues at 4.5 months old. And here is the cigarette smoke machine that actually loads the cigarettes, lights them, puffs them, and blows the smoke into one of these chambers, and the other chamber just gets room air. So again, we have 3 factors: prenatal exposure to cigarettes or room air, adult diet, high fat, low fat, and then sex, male or female, which makes the statistics nice and fun. So here I'm just going to point out a few of the results that we've gotten from this study. At postnatal day 50, that's what PD50 stands for, we're looking at what's called the beta diversity of the microbiome in the cecums of the offspring. Now, a cecum, we don't really have a cecum, but it's kind of like our appendix. So it's right at the beginning of the large intestine, and it's where it's easy to measure the gut microbes that when you collect them. And what we see is when we look at both sexes together, and I should explain what beta diversity is. Beta diversity is basically a measure of the variety of different species present in the microbiome. Okay, so what we're seeing is when we look at both sexes together, what we see is the sham and the CSC animals overlap a fair amount. We still get some differences, but what you really see is when you look at the males versus the females at this age, you see that the males are the primary driver of this effect of cigarettes versus room air when they were in utero, compared to the females that aren't that different. And one of the main groups that we found was different was that there was a decrease in Clostridia, a type of bacterium. And the reason that's important is that Clostridia is important for making the short-chain fatty acid, acetate. So this is all about the cecal acetate at that same age. So I'll tell you a little background. Acetate is a short-chain fatty acid, so it's produced when the microbes ferment carbohydrates that are in the diet that the mouse is eating. So acetate can do lots of different things. It can be a precursor for acetyl-CoA in the TCA cycle that's important for metabolism, promotes protein acetylation, activates an AMPK pathway that's really important for when you're going from fed to fasted. You're in a low energy state, your AMPK pathway pops into gear. It also, so that's what it does inside the cells in these pathways, but it can also act at membrane G protein coupled receptors, and it helps regulate insulin secretion, which is really important for metabolism. So that's acting at the pancreas. It can also stimulate production of a hormone from the gut called glucagon-like peptide 1 that it's released from the gut normally when food hits the gut. And that's a symbol, that's a signal that you're eating and you're going to need some insulin soon. And so what GLP-1 does is it goes in the bloodstream and acts as an incretin. An incretin is a gut hormone that causes increased insulin secretion from the pancreas. So it also is promoting glucose handling, metabolism, metabolic health. And so acetate is involved in metabolism at all of these different levels in a very complicated way. And then you also have acetate acting at those membrane receptors to stimulate leptin at your fat cells, adipocytes, and leptin is also going to have an effect on your metabolism. So what we found here is a little complicated. We're looking at acetate production or acetate content in the cecums of our offspring once they're 50 days old that were exposed to either room air or cigarette smoke in utero. And here we have the males separated from the females. And what these letters up here are indicating is statistically significant difference. So the main thing we see here is that males and females are different. So if you look at the sham male and the sham female, females just have lower acetate levels in their cecums, regardless of what happened with cigarette smoke. What we see here is that exposure to cigarettes prenatally affects the acetate production and presence in the cecums of males. And when you look at everything that acetate does to regulate metabolism, it makes us wonder, okay, well, is there a difference in metabolism with cigarette smoke? And is there a difference between males and females? And the answer is yes. So now we're pulling this out a little bit longer. So remember, at day 50, when we have the cecum data, the animals have been on the same diets. It was at that point we moved some of each sex and each exposure group to high-fat diet and some on low-fat diet. So it's a completely balanced design. And this is a glucose tolerance test. Basically, what you do is you give the animal an injection of glucose and you measure their blood over the course of a couple of hours. And ideally, the glucose should get into the bloodstream because you've just injected it, but the insulin should pop up and should get the glucose out of the blood and into the cells, because that's what insulin does. And what you can see possibly here is that there's a difference. These triangles are the CSC, and they're taking longer to get the glucose out of the blood than the CSC animals. And so what we have done here is area under the curve. So it's basically just taking all of this area and putting it in one bar so that you can look at the difference. And so what we see here is that there is a significant effect in the ANOVA here of gestational cigarette smoke exposure on glucose handling. That basically, in this case, whether you're male or female, if you were exposed to cigarette smoke in utero, you have poorer glucose handling. It takes your body longer to get the glucose out of your blood and into your cells. And that is a model of metabolic syndrome and of gestational diabetes. What we've done here, there was no effect of the high-fat diet in this case, so that has been combined here. Now when we're looking at fasting blood glucose, now instead of area under the curve, we're looking at units, milligrams per deciliter, and now we're seeing that there is an effect of gestational exposure, so an overall effect of cigarette smoke, an overall effect of sex, that males and females are different, and that there's a sex by diet interaction. So the statistics here are pretty complicated, but basically what you can see is that the effect of CSE is greater in the females, regardless of diet. But here, the effect of diet in males, being on a high-fat diet, overwhelms this more than anything else. I think is the most simplest way I can describe what I'm looking at here. So we did lots of other things that I will summarize here that I didn't show you, but basically what we've shown is that prenatal exposure to cigarette smoke in mice alters their gut microbiome in an age and sex-dependent manner. I only showed you one age, but what we see is changes in these microbiomes with age after birth. We showed a decrease in microbial acetate production in the cecums of the males that were exposed to prenatal CSC. We've shown differences in percent body fat, in liver gene expression, in glucose tolerance testing, and insulin levels in a sex-dependent manner. I didn't show the data because I didn't have time, but it affects some anxiety-like behaviors. So it decreases it in one test, increases it in a different test. And we've now also shown that there are some differences in the brain. So the hypothalamus is the part of the brain that does a lot of things, but one thing it does is monitor your adiposity, your how much fat you have, what hormones you have like insulin and leptin, and help dictate your appetite and your metabolism from a brain standpoint. And we have found that the prenatal exposure to cigarette smoke altered these hypothalamic genes in a sex-dependent manner using a technique called RNASeq. So what's next? Well, we still have a lot of tissues and a lot of different ages that we've collected. So we want to follow up that, looking at gene expression and protein expression. We also have, we've got this basic RNA-seq data where we see all of these gene expression patterns being different, but we want to look at what pathways are involved there. And I have been awarded a new grant from the National Institutes of Health, an R15, which is to focus on undergraduate research training. And so what we're going to do now is continue the study to see if those changes in microbiome we've seen are determining the differences in physiology and behavior, because we showed a difference in microbiome, the beta diversity, alpha diversity too, that I didn't show you. We've shown a difference in physiology and behavior. So we showed a difference in glucose tolerance testing and insulin levels and fasting blood glucose, liver enzyme, all that kind of stuff. But we haven't shown that the microbiome difference causes the physiology difference. So now we want to put those two together and see if we can find a mechanistic link. So the way we're going to do that is we're going to do a cecal transfer. So we're going to take the cecal contents of some donor animals that have been exposed to cigarette smoke or room air and cross transfer to recipients to see if changing the microbiome changes the physiology. And then we'll also do this through adding what's called resistant starch. So it modifies the ability of the microbiome to make those short chain fatty acids like acetate. And so we'll then measure all the downstream effects of that modification in terms of physiology and behavior. So it's a lot of work. It's a 3-year grant, and we're just getting started. Actually having a new cigarette smoke machine delivered next week. So all next year or so, we're going to be working on that along with a whole cadre of undergraduate students. This study is in large part done with undergraduate researchers. And so what I wanted to mention just briefly is why you should do undergraduate research. One big reason is to find out how science is done. You learn about science in your courses and you learn what's already been done, but you don't really learn how to do science until you do it. And you also are able to do cutting edge stuff. You are able to not just confirm what is in your textbook like you might do in a course laboratory, but you can do new research that's never been done before and get answers to new questions. In some places, if you're, and I really have focused this on the undergraduates in college, but it's also possible to do some of this in high school as well. But in college, there's something called a senior honors thesis that is basically like doing a graduate thesis on a smaller scale and can help you do things like get the highest honors, like summa cum laude. It goes on your transcript. It's very impressive for your next steps. It helps you explore career options. Most of the biology majors that come into my department come in thinking they are pre-med. They want to be doctors or they want to be dentists. But part of that is just because they don't know what else is out there. So having done some research as an undergrad, they maybe want to consider research as a career, or maybe they want to be a clinician scientist, get an MD, PhD, or in our case here at University of Louisville, we have a very highly ranked dental school where you can also get an MS with your dental degree, the DMD. One other thing that I always mention to undergrads here that are looking to do next steps, whether it's professional school like med school or dental school or graduate school, or even just getting a job, you're going to need reference letters. And if I only know you from class, I know how you are academically. I can write a reference letter for you that talks about that you showed up to class and you asked questions and so forth. But it's really hard to get to know you really as a person just from class. Whereas people who work with me in my lab, I know them as people, and I can tell nice little anecdotes, stories about how they, solved a problem, can talk about how reliable they were, how intellectually engaged they were. I can really get across who they are as a person to an admissions committee that is reading this letter. So that's another good reason to do research as an undergraduate. How do you do undergraduate research? Well, if you're near a university, you can volunteer in a lab. It depends on how old you are, what you are allowed to do. Here, if you're a minor, you might not be allowed to do very many things independently or with hazardous chemicals or with animals, but there are some things you can do. I have had high school students in my lab that did histology and measured, for example, the effects on cell number in a brain region, just looking at the slides without having to touch the chemicals or the actual animals. Once you're in college, you can do research for course credit, which advances you towards finishing your degree. And here we also have summer research programs, and many schools do, where you apply usually in the early spring, and you can be paid to do research over the summer in a lab where you can spend full time learning how to do research and getting a project done that you can present or even possibly publish. At U of L, University of Louisville, here are some of the programs we have. Might not be relevant to all of you, but many universities have similar programs. So there are programs that are around the nation, such as National Institutes of Health, National Science Foundation, USDA, et cetera, that a government agency, sometimes there are private foundations that include funding for trainees. Most of the time, they're going to prefer students that have some experience. So you volunteer first and then get hired once it works out. That's true, especially if you're just going to hang out in a lab at a nearby campus or at your campus if you're an undergrad. However, most of us want people who are going to stick around. So I particularly like to get students working with me when they are early in their programs so that I have them for a while. It takes a while to learn how to do this. And if you spend one semester learning how to do everything and then you graduate and you're gone, I didn't get much benefit out of that. Whereas if you're a freshman or sophomore, you learn what you're doing, and then I still have you working with me for a couple of years, we can get a lot of cool stuff done during that time. So I'd like to acknowledge some of the people who have worked with me in the past. So current students in the CAN lab. I have pictures here of Isaiah Burciaga and Selma Pod Buchanan and Caitlin Chisholm. Some former students such as Megan Hovner and Sophia Brown that did some of the work that you've seen on the graphs that I showed you. Here again, Dr. Rachel Neal, Nika Zabel-Zabels, who are part of the CAN lab, and several other colleagues, including who I mentioned, Deborah Denenfeld with Dancing While the Soldier Project. And of course, I need to acknowledge my funding sources, most of which has come from different branches of the National Institutes of Health. And I would be happy to answer any questions you have.
Hey, thank you so much. That was awesome. A lot of really cool new science for the kids to be exposed to, and it was such a broad range. So I appreciate you taking time to meet with us. I had a question about the undergraduate research, because we do offer a program here at Headwaters called the Headwaters Research Experience, where we do something similar with high school students. You talked a little bit about the value of that, and I know that you did some research when you were younger. So can you discuss a little bit about the value that you have seen in your career from having that experience?
Wow. So one of the programs that I'm involved in is not quite high school, but it's transfer students. So students that are starting at the community college and starting to work in the labs at the university while they're still community college students. And, several of the students have really been encouraged to finish their degrees at the community college, transfer to the four-year school. We're still early in the program, so we have our first people graduating in that program from the four-year university next May. But one of the people in particular came to work in my lab just as one of these summer students, and she was just so good at it. And so we hired her. We had grant money, like from this grant I was telling you about, and we hired her full time, well, part time during the school year and full time in the summer to do research with us. And it's really making her think about what she wants to do with her career. And another thing is that it really gave her a lot of confidence that I think she was lacking. She wasn't quite sure she could do this, and we're telling her, yes, you can. You can do it in the CAN lab. So I think it really, besides boosting confidence and really just showing people what's possible. When I was an undergrad, I was pre-med, in part because I didn't know what else there was. And I ended up changing my mind in part because I realized I didn't really want to be a physician so much as I wanted to understand how people's bodies worked. Mm-hmm. And that led me more to doing research in physiology, because that's how you figure out how you work. And then once I was a postdoc and was working in the field of endocrine disruption, well, that's when I really started thinking more in terms of all of the different things out in the environment and what they do to us that weren't intended. No one made a chemical intending it to mimic estrogen to be out in the environment, to make fish, boy fish turn into girl fish, for example. But that is what has happened just because science is like that. Physiology, all these vertebrate species have so much in common, like we mentioned before, with the birds and the rats. They're very different evolutionarily. They don't look the same, they don't act the same, but the chemicals in their bodies are very similar. And so when there is a chemical out in the environment that affects estrogen receptors of a fish, it very likely affects estrogen receptors of a cow and estrogen receptors of a person, because we're all so similar. We all have so much in common once you get down to the cellular level. And I kind of went way off of your question, but that was kind of my path from just thinking I want to be a physician because I don't know what else there is, to really realizing what is it about biology that I like, to then realizing how what I'm doing in biology could have implications for human health and environmental health.
No, that's fantastic advice, because you just never know where you're going to end up. And for every question you think you might have answered, there's going to be 20 more. So I think that's perfect advice to give to the students that are going to be watching. So thank you.
You're welcome.
Yeah, so with that, I just wanted to thank Dr. Corbitt for taking the time to meet with us today and sharing all of her work and the awesome advice that she just gave on doing research when you're younger and getting all that hands-on experience and start networking now. So if that's something that you would be interested in doing, like I said earlier, we do have a research experience program that is very similar to what was just described. We have kids from 8th grade all the way to 12th grade that do their own individual research with a mentor, and we even publish you. So if you're interested in checking that out, please look at that link below. If it's easier for you or you have any questions, you can go ahead and just email me at jen@headwaterscienceinstitute.org. So again, I just wanted to thank you so much, Dr. Corbitt, for sharing your knowledge and your experience with us. And again, appreciate you helping.
You're welcome. I enjoyed it.
Thank you. Want to learn more about our Lunch with the Scientist program? Make sure to like and subscribe and visit us online at www.headwaterscienceinstitute.org for a full list of our video library and curriculum.